Hello, welcome back to Introduction to Genetics and Evolution. In the previous videos, we talked about some of the fundamentals associated with natural selection. I reintroduced that it is a mathematical inevitability. We talked about the three conditions for it, and we looked briefly at changes in allele frequencies and genotype frequencies associated with the action of natural selection. Now, one point I stressed in the last video was the importance of dominance. That dominance affects how natural selection shapes allele and genotype frequency. Now, in the last example I showed you in the last video, we had all adult little a individuals die. So we said that at age 10, all little individuals just die off. Let's follow up on this with a couple of different examples. I'm going to change, in this case, the alleles from big A and little a to M and N. And that way there's no implication of capital being dominant or recessive or anything like that. So let's say now we're looking at the relative fitness of three possible genotypes. Again, there's two alleles. Genotype MM has a fitness of 1.0. This is again relative fitness. Genotype MN has a relative fitness of 1.0. And genotype NN has a relative fitness of 0. As in all individuals that are NN are dead. Now, which allele is dominant? Which allele is recessive? Well, we define dominance by what the heterozygote has the same phenotype as. The phenotype we're looking at in this case is the relative fitness. So which homozygote does the heterozygote have the same relative fitness as? In this case, the MM homozygotes have the same relative fitness as the MN heterozygotes. In this case, this must necessarily be dominant, this must necessarily be recessive. Okay? Because these two have the same fitness, they must be this one must be dominant, the M allele. This one is recessive. In this particular case, the recessive one is also bad. It's the one that has the lower fitness too. That won't always be true, but in this particular example, that is true. So N is recessive to M, and N is detrimental or bad. There's a different example. So in this case, MM has relative fitness 1.0, MN has relative fitness of 0, and NN has relative fitness of 0. So what do we think is going to happen in this case? Well, first of all, which one is dominant? In this case, again, we look at the heterozygote. Which one has the same relative fitness as it? In this case, it is the NN homozygote. So N is actually dominant now. N is still bad. So in this case, the dominant one, this one is dominant, N is dominant. And what will selection do differently? Well, we might guess in this case that selection will actually be much more quick. Because in this case, you're eliminating all N's from the population at once, instead of just a subset of N's from the population. Here, some N's are sheltered. Here, no N's are sheltered. They are all exposed to natural selection. So, with a dominant detrimental, the heterozygotes will also respond to selection. Okay? Now let's do a third example. Here we have MM is 1.0 for its relative fitness, MN is 0.5, and NN is 0. In which case, which one is dominant? Well, the simple answer is neither, because no homozygote has the same relative fitness as this particular heterozygote. So we can call this a case of no dominance. In all three of these cases, we assume that the NN heterozygote is the worst one. This is what's often referred to as directional selection, because selection will preferentially push towards the elimination of the N allele over time, whereas the MM is always perfectly healthy. But the rate will be very different. In this case, selection would be very slow to eliminate N's. In this case, selection would be very fast to eliminate N's. In this case, it would be intermediate. Now, we can see this. Here is the effect of dominance with directional selection. This is using that allele A1 software that I mentioned before. So let's look at the case now. The alleles in this case are called A1 and A2. So if we have allele genotype A1A1 has a fitness of 1.0, genotype A1A2 has a fitness of 0.5, so we're not eliminating all of them, we're just eliminating half of them. It has half the relative fitness. Genotype A2A2 has a fitness of 0.5. This would be a case of a dominant detrimental. We say it's dominant because, again, you look at the heterozygote, it has the same fitness as this one. Right? So A2 is dominant, A2 is bad. A2 will be going away from the population. In this case, since no A2s can hide, it goes away pretty quickly. In contrast, when you have the recessive detrimental, now this one is dominant, A1 is dominant, but A2 is the bad one. So you can eliminate these pretty readily from the population, but these are perfectly okay. So as you can see what happens here, if you look at the change over time, the x-axis here is time in generations. The y-axis is the frequency of the A2 allele. You see the A2 allele is going down in frequency, but it's going down somewhat gradually as it gets near the end, because a lot of these individuals, when you're down here, a lot of the A2s are hiding as A1, A2. Whereas over here, none of them can hide. They're immediately exposed to selection. Boom. Whereas here, the A2s can stick around, and therefore you maintain some A2 in the population over a fairly long period of time. Now, in fact, this is actually what happens for many bad mutations. As I mentioned a long time ago when we were talking about inbreeding depression, many bad mutations are either completely or mostly recessive. Some examples of genetic diseases like this are Tay-Sachs, cystic fibrosis, etc. They're maintained in the population by carriers. Selection is inefficient at getting rid of them. Now let me give you a problem to try. Which of these depicts the change in lactose intolerance allele? This is the big A that we talked about in the previous lecture by selection. So I've given you here the relative fitnesses. So big A has a relative fitness of 0.95. That is when it's lactose intolerant. That's also the ancestral type. Big A little a has a relative fitness of 1.0. And little a has a frequency of 1.0. Or I'm sorry, a fitness of 1.0. Again, these pictures are showing time and generations on the x-axis and the frequency of big A, make sure you know that, the frequency of big A on the y-axis. Which of these would be an accurate depiction of what we expect to see over time? Well, I hope that wasn't too difficult. Now, what do we have here? Now, which allele is dominant? That's the first question to ask yourself. Well, the allele that's dominant in this case would be little a, because it has the heterozygote has the same relative fitness as the little a homozygote. So this one is recessive. Big A is recessive, and it's also bad. So we have a case of a recessive detrimental that's being eliminated from population. What we're looking at in this case is the frequency of big A. So that is the recessive bad allele. Since it's bad, we know it'll go down in frequency. So we can immediately eliminate these two. Since we know that big A is bad, it should go down in frequency, not up in frequency. Now the question is, does it go down fairly quickly, or does it do this very slow thing? Well, as I mentioned in the last slide, when it's recessive, a lot of individuals here can hide as big A, little a, and it'll be slow. So in this particular case, number 1 is the correct answer. Now again, there are many types of selection that can happen. And there's in fact many types of what is referred to as directional selection. And this is what we've been looking at so far. In directional selection, one allele eventually replaces the other. That everybody in the population eventually, if you wait long enough time, will be either big A, or little a, little a. So what you have is something like this, where the fitness of big A is less than or equal to the fitness of big A, little a, is less than or equal to the fitness of little a. Or the opposite, where the fitness of big A is greater than or equal to the fitness of big A, little a, is greater than or equal to the fitness of little a, little a. Now the examples can vary. So here we have a case where we have a recessive detrimental for the first line. We see this because the dominant one has the high fitness, right? The recessive one. So eventually all the population will be big A. All right, here, what's happening eventually. Okay. In this particular example, again, everybody eventually will be big A. This is a case of no dominance, or intermediate dominance, as some people call it. In this particular example now, we have little a has higher fitness than the other two. So eventually everybody in the population will be little a. And here's the example of lactase persistence or lactose intolerance, that eventually everybody in the world, if we wait long enough periods of time, everybody should be little a or lactose tolerant, or lactase persistent. Now this may not happen in real life for this particular example because we now can purchase lactase over the counter, but that's the direction that things have been. So these are all cases of directional selection where eventually you go to one form. You eventually go to monomorphism. Here is a different type of selection. This is the case of heterozygote advantage. This is often called overdominance. In that particular case, the most fit genotype is the heterozygote, or the big A, little a. So this one is more fit than both kinds of homozygotes. The two homozygotes may differ in fitness from each other, but the highest fitness is associated with the heterozygote. Okay? In this case, unlike with directional selection, one allele does not replace the other. This does not lead to monomorphism over time. The classic example of this that people always cite is that of sickle cell anemia and malaria resistance. So let me introduce this to you briefly. Malaria is a big threat for a large part of the developing world, In fact, in places like sub-Saharan Africa, your odds of death by malaria are around 4%. That's pretty high overall. Now, malaria is transmitted by mosquito bites, which are giving this Plasmodium protozoan, which is what actually causes malaria. Now, how does this relate to sickle cell anemia? Well, sickle cell anemia is not a great thing either. This is a recessive genetic disease, so only little a individuals are fully afflicted. In this particular case, the sickle cells die faster than normal red blood cells, and they deliver less oxygen to cells. So people who have this disease have, are tired all the time. They have chronic pain. They have quote-unquote episodes where they sometimes have to go to the hospital associated with this. So this is clearly not a good thing. Interestingly, the heterozygote, so the big A little a's, these people are often called having the sickle cell trait. They're usually okay. Sometimes they can have some sickling of their cells when they've exercised very hard. They've had some sort of intense physical exertion. But overall, they're good. But interestingly, these individuals that are heterozygous for sickle cell are actually more resistant to malaria than individuals that are homozygous for the non-sickling allele. Now, the thought a long time ago of this was that invasion, growth, and development of Plasmodium was actually somehow reduced in these heterozygotes blood cells. But more recently, I cite two studies down here. You're welcome to look them up if you'd like. One thought is the heterozygote is more tolerant to sickle cell symptoms, but actually still retains infection. Whereas another study showed that the infected heterozygote cells are more likely to be eliminated by the spleen. So there are a couple of different explanations for this, but the important thing is that if you're in a place where there is malaria, if you're a heterozygote, you are actually not immune, but you're much less likely to get it than individuals that don't have sickle cell trait. So the sickle cell exhibits a heterozygote advantage in some populations, because you can think of it this way. The big A's are susceptible to malaria, so they might have a relative fitness like 0.85. I'm just making up these numbers in this case. The big A little a's are generally fine, right? They don't really get malaria, they don't have sickle cell disease, so they'll have the highest relative fitness, 1.0. And little a's have the sickle cell anemia disease, which is extremely debilitating. So let's say their relative fitness is something like 0.05. Well, what would be the fate of the little a allele if it arose as a mutation in a big A population? Well, it seems like little a is really bad. On the other hand, big A little a is pretty good. So what should happen? Well, we can simulate this using allele A1. And we see that the frequency of little a rises and then comes to this equilibrium. It comes to this stable allele frequency and it just stays there. Now, the number it comes up with in this case is the frequency of little a, or A2 in this case, is 0.136. That's what was estimated using allele A1. Now, interestingly, we can actually apply the math ourselves and see what happens. So the frequency of little a, this is the prediction from what you should see in terms of changes in allele frequencies in the equilibrium allele frequency. So this is at equilibrium. A little hat over this to indicate equilibrium. At equilibrium, what should the frequency of little a be? Well, we'd say it's 1 minus the relative fitness of big A, divided by 1 minus the relative fitness of little a, plus 1 minus the relative fitness of big A. Well, we have all these numbers here, right? So the fitness of big A, this one is 0.85. The fitness of little a, 0.05. The fitness of big A again is 0.85. So we can substitute these numbers into this very simple formula. So there we go. They're put in there. And this calculates out to 0.136. That number may look familiar to you because that was exactly where we saw this equilibrium drawing here. So the important thing, this is actually a stable equilibrium. That essentially, if you're not at the equilibrium allele frequency, selection pushes you towards it. If you're above it, you go down to it. If you're below it, you go up to it. You're drawn to this equilibrium allele frequency. And again, this is called overdominance. Both alleles are retained, and if you're not at the equilibrium frequency, you will move to it. Now let me show you a graphical representation of that. Here it is. Here's a graphical representation of changes in allele frequency associated with overdominance. On the x-axis here, I have the frequency of the big A allele, and on the y-axis, I have the predicted change in frequency of the big A allele in the next generation. Don't worry about this too much if you're not exactly following, but this just depicts the same sort of thing. So imagine that you're at, let's say in this case, the predicted allele frequency, equilibrium allele frequency is 0.6. If you're at 0.4, if you're at the actual frequency of 0.4, your predicted change in frequency of big A is what? In this case, your predicted change is positive. So I'll push you towards the equilibrium. If you're over here at 0.8, your predicted change in allele frequency is negative. So that pushes you in this direction. So you can see anytime you're anywhere but at the equilibrium frequency or where you have no variation, then you will always be drawn to that equilibrium unless you're at either 1.0 or 0.0 where there's no variation. Again, selection requires variation to act. So this is just a graphical representation of what I told you in words, so don't worry about that. Again, the important thing with heterozygote advantage or overdominance is there is this predictable equilibrium allele frequency that the population will be drawn to. Now let's talk about the opposite, heterozygote disadvantage. What happens if the heterozygote has the lowest fitness? There aren't very many good cases of this, but we can at least show mathematically what should be expected. Well, in this case, again, the least fit genotype is the heterozygote, so I just made up some numbers here. What this does, this actually leads to an unstable equilibrium, kind of like this fellow standing on the chair here. That if you start below the equilibrium allele frequency, so again, the equilibrium allele frequency is 0.27, so you're just a tiny bit below it there. If you start below it, you just go away completely. You lose one allele completely. If you start above it, you lose the other allele, and one allele goes to fixation. It's only if you're right at the equilibrium allele frequency that both alleles can persist. But in a real population, you probably can't do that because unless you have an infinite population size, you can't stay exactly at that allele frequency. So unlike the previous example, this is a graphical representation of allele frequency change with underdominance. If you're not at the equilibrium point, let's say, for example, again, the equilibrium allele frequency is 0.6. If you're starting at 0.8, again, your predicted change allele frequency is positive. You get pushed away. If you start at 0.4, your allele frequency predicted change is negative. Negative. You get pushed away. So again, you lose variation in the population. Only if you're right at that equilibrium allele frequency can it stay. So it's a very unstable equilibrium point. It probably doesn't happen very much. Or if it does, it's eliminated so quickly that we never see it. Okay? So those are three kinds of single locus selection we've talked about. Directional selection, where one allele is just consistently favored. Overdominance, where variation is retained in a stable way. Underdominance, where variation can be retained temporarily in an unstable way, but eventually you lose variation, and it's unpredictable which one you would lose unless you know that you're above or below the equilibrium allele frequency. There's one more type I was going to introduce, and that is that of frequency-dependent selection, and specifically negative frequency selection. Now, the previous examples all assume that fitness was independent of what was going on in the rest of the population. That, you're big A, you're better than big A, little a, no matter what, right? Or if you're little a, you're better than big A, no matter what. Sometimes it's actually better to be rare, that your fitness may depend on your relative abundance or frequency in the population. Now, being better makes you more common. Right? That if you're better, it means you're having more kids, you're more likely to survive, so you become more common. But if it's better to be rare, it's kind of this funny thing where you have these opposing forces. What this does, and this is the case specifically of negative frequency-dependent selection. We're not going to talk about positive frequency-dependent selection. If you have negative frequency-dependent selection, this assumes that it's better to be rare, but being better makes you become more common. This eventually leads to an equilibrium. Let me give you an example. Let's use the case of sex ratio. So in many species, sex is determined genetically, as you know. That in mammals, we talked about the sex chromosomes, that XY is what causes maleness. And you're kind of locked into this by transmission. But there are other species where alleles at one gene can cause an individual to become a male versus a female. That you can have this sort of variation within it. Now, if females are very rare in the population, and you need both males and females to make offspring, is it better for you to produce male or female offspring? Is it better to produce the abundant type? Or is it better to produce the rare type? Essentially, in this particular example, would selection favor the male or the female allele? It's probably better to produce the rare type because you're more likely to mate. If you produce the common type, you're very likely not to mate. When you're rare, this rare allele has an advantage. As it becomes more and more common, it loses its advantage and eventually can go to a disadvantage. This will maintain genetic variation in a population. And you might guess, if you have two alleles, what would you predict that equilibrium allele frequency to be, if you assume it's symmetric? Well, the answer is 50%. In fact, if it's symmetric, you will tend to see just about always the equilibrium frequency be 1 over the number of alleles. So if you have two alleles, it would be 1/2. What if you introduced a third allele into the population? well, then probably that equilibrium allele frequency would be 1/3. This is assuming a sort of symmetry, which may or may not be accurate, but this gives you some idea of what it could be. Now, frequency-dependent selection is another way of maintaining variation in a population through selection. And we talked about a couple of different modes of single locus selection here. Some of them remove variation, some of them retain variation. Okay, so directional selection favoring one allele, this essentially eliminates variation, right? And the effects of dominance will change the speed with which variation is eliminated, as well as the strength of selection, of course. Heterozygote advantage maintains variation. Heterozygote disadvantage is unstable. So although you can argue that it maintains variation, it only does so under unrealistic circumstances. So it probably will eventually lead to elimination of variation. And negative frequency dependent selection will maintain variation as well. All of these are affecting genotype and allele frequencies, but they're acting through the phenotype. Now, this discussion, we're looking at everything in the context of the effect of alleles at one gene. Obviously, that's not what Darwin did. And it's obviously not what people do when they're applying artificial selection, right? They're looking at the traits as a whole. They're looking at the phenotype, the variance. So let's tie this back to the principles we talked about earlier in terms of heritability and things like that. Well, we'll start doing that in the next video. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we looked at the types of selection that can operate on a single locus or single gene in the genome. In that case, we looked at the effects of dominance, and in particular on directional selection. We talked about other forms of single locus selection, such as heterozygote advantage, which is overdominance, underdominance or heterozygote disadvantage, and negative frequency dependent selection. Now, if you go back, Darwin, of course, didn't know anything about genetics. He instead thought of natural selection in the context of variation in traits. And that's what a lot of breeders do when they're applying artificial selection to, say, their dogs or cattle or corn or whatever they may be using. Now, natural selection can be studied in the context of quantitative traits or in the context of single loci. Now, when we talked about in the context of heritability previously, we were thinking of it in terms of these quantitative traits. And you don't necessarily need to know the underlying genes to infer what kind of selection is operating. So let's talk about the types of selection that can be inferred when you're looking at phenotypes and changes in phenotypes over time, or changes in traits over time. This is a picture from the Freeman and Heron book, and this depicts 3 kinds of selection that I'll introduce to you. Directional selection. This is a little bit different than the previous directional selection we talked about when we were looking at a single gene thing. It's very unfortunate they happen to have the same name. But in this case, we're looking at selection which favors one extreme of the distribution over the other. So we have here in the top picture the number of individuals before selection, so it's a particular distribution. We have in the second graph the probability of surviving. So in this case, the ones that have the higher trait values have a higher probability of surviving than the lower ones. And then in the bottom part, we see the number of individuals having these different traits after selection. In this case, we see sort of a change in the mean. The mean here started just below 7. Here it's just above 7. So this would be directional selection. I'll introduce each of these in more detail in just a moment. We have one called stabilizing selection, where individuals with the intermediate value are favored. And we have disruptive selection, where individuals with the extreme values are favored. Let's go over these in terms. So this first example is directional selection, and we're applying this in the context of phenotypes. We're not looking at the single gene version of it, and I apologize again, they tend to have the same name, which is very confusing. But again with this, individuals at one end of the distribution are favored. So if we imagine the x-axis here is height, let's say that's the trait value on there, then with this particular thing, then the probability of surviving is higher for those that are taller, whereas it's very low for those that are short. So what this will do is it'll change the mean of the population over time. The assumption here is that there's some heritability, that there's some heritable component to height, and therefore selection can induce this sort of a change. In this case, what you see is you see the shift over from a mean of just below 7 to a mean of just above 7 in this fictional example. Well, let me show you a real example. This shows change over time in pink salmon weight in Canada. So this is from 2 different places where pink salmon were caught several times. We see here that Average weight in 1951, 1952 was a little bit over 4 pounds, and we see over time smaller and smaller salmon are caught. Same thing with the Upper Johnston trait. Over time, smaller and smaller salmon are caught. Now this has been shown in pictures as well. Here's an excerpt from a magazine. This is showing collections from the same place over 3 time periods. Here's the 1950s. Look at the size of those fish. They're mammoth. The 1980s, they're pretty good. And in 2007, they're fairly small. What happens? Well, when people go fishing, they go out there, they catch the biggest fish, and those are the ones they take home and eat. Smaller fish are less likely to be caught, or maybe more likely to be thrown back. So over time, there's been this selection for favoring smaller fish, or for smaller fish. That is directional selection acting on the phenotype of size, in this case in pounds. Well, let's look at an example of another kind of selection. This is an example of stabilizing selection. Okay? This is the case where individuals in the middle of the distribution are favored, so they have the highest fitness, whereas individuals near the ends have a lower probability of surviving. In this particular case, there's no change in the mean, but you actually end up losing some of the extreme. Well, there's a classic story that goes with this. In the late 1800s, a fellow named Bumpus lived up in New England, and he was interested in the sparrows that were found there. A big ice storm hit New England. Lots and lots of birds died. He went out there and brought back a lot of the birds that were sitting out there in the snow, not doing so well. And what he found, looking at these house sparrows, he brought in 136. 64 of them died and 72 survived. What he found is that the mean of the survivors and the mean of the dead was about the same. Right? They were about 26 grams in size. In contrast, if you look at the extremes, all the ones that were more than 28 grams and all the ones that were less than 23 grams died. There were some that died that were intermediate there, but all the ones with extreme values died, so they had a lower probability of survival. Whereas the ones with the intermediate traits, they were more likely to survive. Well, there's a definite example of stabilizing selection that operates in humans. Can you think of what that would be? Something where the intermediate values are favored. Well, that answer is human birth weight. That again, when we tend to think of very small babies that are 2 or 3 pounds, their probability of survival is not very high. You know, an 8-pound baby, 9-pound baby, they have a pretty good probability of surviving. But when you get up to the 12, 13-pound babies, again, it goes back down. That, you know, a lot of them do survive, especially with medical intervention, but the probabilities tend to go down, especially if you have less medical intervention there. Again, individuals in the middle of the distribution are favored by natural selection. So this would be a case of stabilizing selection. I should emphasize, stabilizing selection is not the same as overdominance. Overdominance is something that happens when you're looking at a single gene. This is looking at a pattern in phenotypes. It's a common error. I just want to make sure to emphasize that. Last but not least, disruptive selection. This is basically the exact opposite of stabilizing selection. That individuals at the 2 ends of the distribution tend to be favored. Again, in this case, you have no change in the mean, even though you're losing those individuals that may have the mean values. But you have an increase in those with the extremes, both high and low, and you have a loss of the intermediate phenotypes. So you have basically a greater variance over time. There are examples of this. This is one that was published in the around 1992 or so. This is looking at beak size in African finch. And we found that, and what people found is that when you're looking at the females, they tend to specialize on 2 sedge species seeds. And those with particular bills, this set can specialize on one type, this set can specialize on another type. But the intermediate ones actually are not as good at either type of seed. So the intermediate ones here, the ones near the mean, are not as good. They have lower fitness. It is better to have one of the more extreme phenotypes. So those are, again, the 3 types of selection that you can infer from looking at phenotypes. Directional, where you have a change in the mean and one end is favored. Could be the other way too. Stabilizing selection, where the ones near the mean are favored, you lose variance, you lose those extremes. Or disruptive selection, where the intermediate ones are unfavored and you have an increase in variance, an increase in those extremes. Now, before we leave selection behind for this particular lecture, I'd like to close with a few final thoughts. So, is selection good or bad for a population? We tend to conceive of selection as being something bad. We tend to imagine this sort of struggle and some genotypes not being able to pass themselves on, some individuals being left out of the population, whatever species it is, we're looking at animals, plants. In fact, that's a very biased view. Natural selection is preferentially reducing or eliminating the bad genotypes. When I say bad genotypes, it's those ones that are not as well suited to passing along or producing offspring, right? So either they have a deficit in survival or they have a deficit in reproduction relative to other genotypes. As such, because we're getting rid of these so-called bad genotypes, the average fitness of all the remaining individuals in the population after selection actually goes up. That if you look at the people who are still there after selection's operated, on average they tend to be more fit. And the reason for this is that the bad alleles have been removed. And this is especially true when you're looking at simple directional selection. And ultimately this will lead to a long-term improvement of the population's ability to survive and reproduce. Now, in this regard, directional selection is improving the average population fitness, and the speed with which it does it has been described by Ronald Fisher, who's depicted here on this slide, in what's referred to as Fisher's fundamental theorem of natural selection. This is elaborated very nicely in his 1930 book, but he identified that the rate of increase in fitness under directional selection is equal to the genetic variance in fitness. And we could think of this as similar to the heritability aspects we talked about earlier, right? Selection acts on that aspect of the trait that has a genetic basis, right? That VG is what responds to selection out of the overall VP. So in terms of overall fitness, we see the same sort of concept applying. The rate of increase in fitness is equal to the genetic variance in fitness. So if there's a lot of variance in fitness, then you'll see large step changes in the overall average population fitness. In contrast, if most individuals have fairly similar fitness, you won't get that much of an increase in overall population fitness. So I just wanted to present that to you as a couple of closing thoughts because these are some neat ideas that have been around for quite some time. And I hope you enjoyed these lectures on natural selection. In subsequent videos, we'll look at another very important evolutionary force, but one that's far more random. We'll be looking at genetic drift. So stay tuned. Thank you.